The Krebs cycle from grade 11. The Krebs cycle comprises of mainly 7 steps, and it is also called the citric acid cycle or the tricarboxylic acid cycle. Firstly, the pyruvate formed by glycolysis, that is a 3-carbon compound, gets combined with coenzyme A to form acetyl coenzyme A, which is a 2-carbon compound. The acetyl coenzyme A ultimately enters into the Krebs cycle. The first compound that is formed is citric acid, and it is a 6-carbon compound. The citric acid then changes into alpha ketogluteric acid, which is a 5-carbon compound. The alpha ketogluteric acid then converts into succinic acid, which is a 4-carbon compound. The succinic acid changes its form and turns into malic acid, which is again a 4-carbon compound. The malic acid again changes its structure and changes into oxaloacetic acid, again a 4-carbon compound. Then the oxaloacetic acid again regenerates and forms or combines with acetyl coenzyme A to form citric acid. So this is what you should keep in mind first. The pyruvate changes into acetyl coenzyme A and then further it enters into the Krebs cycle. Now, if you know all of these steps, then learning the Krebs cycle is going to be really easy. Starting with the conversion of pyruvate into acetyl coenzyme A. The first thing that you'll notice is the reduction of a carbon because it is 3-carbon and here it is 2-carbon. So easily you can write it is decarboxylated, or one molecule of carbon dioxide is released when it was converted, that is pyruvate was converted to acetyl coenzyme A. But where did this coenzyme A come from? So again you can know that coenzyme A was added to pyruvate to form acetyl coenzyme A. So coenzyme A was added, carbon dioxide was removed. After that, another thing you should know that since there is decarboxylation, there was a loss of energy or a production of energy. So again you can write that NAD plus was converted to NADH plus H plus. Okay, so the 3-carbon pyruvate was converted to 2-carbon acetyl coenzyme A. For that, coenzyme A was added and carbon dioxide was reduced. And when this was changed, the NAD plus turned to NADH plus H plus. This acetyl coenzyme A is combined with oxaloacetic acid. See, 4-carbon plus 2-carbon gives us 6-carbon. So acetyl coenzyme A is combined with oxaloacetic acid to form citric acid, which is a 6-carbon compound. Now again, if you look here, we can see there is a reduction of 1 carbon molecule. So again, you can write there is decarboxylation, so release of 1 carbon dioxide. Again, since there is decarboxylation happening, there should be a release of energy. So once again, NAD plus was converted to NADH plus H plus. This is how the cycle goes on. Next, again, when you look, you can see that this 5-carbon compound is changed to 4-carbon compound. So again, once again, the alpha ketogluteric acid releases carbon dioxide to form succinic acid, which is a 4-carbon compound. And when carbon dioxide was removed, there was a release of NAD plus to NADH plus H plus. Now one thing that you need to keep in mind is there is also a change of GDP to GDP. This is the state or this is the stage when ATP was made. So this is where ATP is made in the Krebs cycle. For isomerization or to change the structure into something else, you use FAD plus and converts it into FADH2. Whatever I'm telling is a trick to help you learn this. If you learn it in this way, this is going to be really easy. And then finally, again, malic acid is isomerized to oxaloacetic acid. And again here, NADH plus is formed from NAD plus. So once again the cycle, pyruvate is converted to acetyl coenzyme A. Coenzyme A is added, CO2 removed, and then NADH plus was formed. Next, acetyl coenzyme A and oxaloacetic acid combined to form citric acid. When citric acid was converted to alpha ketogluteric acid, carbon dioxide was removed, NADH plus was formed. Then alpha ketogluteric acid was converted to succinic acid. There is a reduction of carbon dioxide, and then NADH plus was formed, as well as ATP was formed. This is really important point. After that, succinic acid was converted to malic acid. It was isomerized, so we used FAD plus to convert it into FADH2. Then malic acid changed to oxaloacetic acid, and NADH plus was formed. And then another thing that you need to keep in mind is that 1 NADH2 gives us 3 ATP and 1 FADH2 gives us 2 ATP. And everywhere here we used 2 NAD plus, as well as here 2, here 2, FADH2 also we used 2, and then NADH also we used 2, as well as GDP. GDP, the full form of GDP is guanosine diphosphate. Guanosine diphosphate. Guanosine diphosphate. So I hope Krebs cycle is picture perfect for you. You just need to understand what the cycle is about, and the rest will be easy. Learn about where, and then remember where a decarboxylation is happening. Those will be asked for exams, as well as where all NADH plus was formed and where is ATP formed. They won't mention anything about GDP and GTP, only about ATP. So if this helped you, please like and subscribe to the channel and follow for more. Thank you.